So we can go from a mutation to an illness in just 4 steps. The first thing that would happen is the mutated gene codes for a non-functioning protein, usually in an enzyme. Enzymes make every process in our body happen faster so that we are capable of life. And then the non-functioning protein, non-functioning enzyme can't catalyze the reaction as it normally would, bringing it to a halt. So imagine if you have a mutation that makes it so that you don't have amylase in your saliva. You can't start that digestive process as your food is moving down your body. If you can't get the right amount of energy out of your food, then how are you going to be able to survive? You need to have that energy in order to be able to go through all of your daily processes. So the third step would be the molecule at which the enzyme would have reacted accumulates like a blocked assembly line. So if your food remains undigested in your body and you can't get the energy from it, then imagine that if you're not getting the energy from your food, you're not getting the energy to survive. If you think about somebody like me who has diabetes, if I don't take my insulin every day, then I'm not able to get that energy from my food, and my blood sugar gets really high. And after a while, as I can't get that energy from my food, it can cause all kinds of problems in my kidneys, in my fingers and toes, in my brain, and eventually it could end up killing me. So the accumulating chemical causes sickness and or death. So just that mutation in my pancreas that made it so that my pancreas doesn't work anymore and doesn't produce any insulin. If I don't have that insulin, that enzyme that helps me to digest my food, if I don't have it, then it's like a blocked assembly line. If you cut off one part of the assembly line, you don't get the finished products, then you're in deep trouble. So the next thing that we're going to move on to is biotechnology. And biotechnology has been amazing as far as producing improvements in agriculture, but it's also been somewhat controversial because some people believe that we're coming up with Frankenfood, that we have foods that are not working the way that they're meant to. And therefore are genetically modified foods that people don't want to eat. So if you hear about the organic movement, that's what this has to do with. So what is the basis for biotechnology? First, we have genetic engineering, which is basically adding, deleting, or transplanting genes from one organism to another to alter the organism in useful ways. So for somebody like me who has diabetes, I have gotten my human insulin through genetic engineering. So the way that human insulin is produced is not by taking it from the pancreases of other humans. It's actually produced by a bacteria in a huge vat, and the way they've done that is by making these bacteria, adding in these DNA sequences that make it produce human insulin. Used to be back before human insulin, and when I was first diagnosed with diabetes in 1985, all of the insulin either came from a pig pancreas or from a beef pancreas. And so part of the natural process of making bacon and ham and all that good stuff was taking the pancreas and removing the insulin from it, and that's what kept diabetics like me alive for many, many years. And then with the advent of biotechnology, they were able to make these bacteria that could produce insulin that was identical to human insulin. And so obviously human insulin is going to be better for me than something that is not the same species, like a pig or a cow. And so this advent of human insulin is really amazing for allowing diabetics to be able to get insulin that's more like the insulin that their body would produce. So the way that that's done, and if you picture your bacteria that has the circular DNA inside of it, and that's what the picture is in the background. If you hadn't guessed that yet, it's DNA. So basically, chop up the DNA from a donor species that exhibits a trait of interest. So imagine that we take cells from people that have healthy pancreas, and we take out the section of DNA that codes for normal, healthy human insulin. So then in amplification, if you're a stereo buff, you can think of amplifying sound, making more of sound. This is making more DNA from very small samples of DNA, so making lots more of it. Then taking that piece of DNA and inserting it into bacterial cells or into viruses, growing the separate colonies of bacteria or viruses, each containing some donor DNA. So like I said, these big vats of human insulin are being grown in these big vats of bacteria. Then finally identifying colonies of bacteria or viruses that have the DNA for the trait of interest. So in going through biotechnology, they don't necessarily need to use every one of those steps. Sometimes one or two are used, sometimes all five are used, but it really depends on what you're trying to do, what you're trying to fix, what you're trying to heal, what you're trying to produce.